Hello, my name is Nathan Rockenbach, and this presentation is about Alphafold, a predictive model for protein structure created by Alphabet. Alphafold was originally conceived as a way to solve a variety of problems in protein structure analysis. Traditional structure analysis methods are time-consuming, taking years and costing thousands of dollars. While considered accurate, these methods are simultaneously considered a choke point in the study of life and the development of drugs and treatments. Alphafold was created to alleviate this cost in both time and money and allow these advancements to be accelerated. To understand Alphafold, one needs to understand how it was created. Alphafold is set up using machine learning. This method is based off of teaching a program by feeding it input data and allowing it to create associations based on that input data. In Alphafold's case, this input included the protein structures already discovered and their amino acid chains. From this, the program could recognize patterns and use associations to build complex structures out of sequences. These outputs are then considered the theorized protein structures until experimental standards could determine the actual structure. Some of the more traditional methods, such as crystallography and NMR, have both allowed understanding of protein structures in a pre-bioinformatics world. These methods require analyzers and interpretation of data to generate the likely shapes of proteins, which can then be adapted into the computer-generated images we usually see when talking about protein structure. In these methods, the analysis is a long and drawn-out process with room for trial and error. While these methods are time-consuming, they have given the base of understanding used as a jumping-off point for AlphaFold. As previously alluded to, the base of structural knowledge functioned as the inputs for AlphaFold's training, which in turn allowed it to start giving predictions. These protein inputs could be divided down into subcategories. Basic structures, such as the commonly studied alpha helix or beta sheets, were determined and input as established protein structures, along with their corresponding amino acid sequences. Similarly, many single subunit proteins were determined and input with their sequences in the same way. It was theorized that if all subunits and structural patterns could be determined, more complex proteins, consisting of multiple subunits, could also be determined by assembling the basic parts in different ways. As it turns out, this was somewhat the case. Alphafold can assemble these different subunits via its pattern association programming to allow relatively accurate protein structures to be generated. As when any newly developed technology is created, the accuracy of Alphafold was tested. In both 2018 and 2020, Alphafold was deemed the winner of the Critical Assessment of Protein Structure Predictions Conference. In both years, Alphafold was deemed a highly accurate alternative when put up against other predictive methods for structure analysis. Alphafold was considered so accurate that it actually caused the criteria of future events to be significantly altered. At the 2022 event, judges were no longer looking at basic criteria like model accuracy or refinement, instead adopting a more complex criteria such as predictions of incorporated RNA structures or protein ligand complexes. The predictive accuracies were mostly seen in areas that correlated to similar structures in well-studied proteins. This accuracy discrepancy was likely due to the nature of machine learning, having predicted outputs based on the already determined inputs. With less correlating input, less predictable outputs would be given. This is actually acknowledged in the Alphafold database when looking at predictive structures. Ranges in predicted accuracies, from very high confidence down to very low confidence, are shown by color on the predicted structures when visualized. Alphafold can be put to many different uses. The obvious use is for the determination of unknown protein structures. While predictive in nature, it can help researchers draw conclusions from experimental data. Alphafold could be used to determine the effect of SNPs, or other mutations and evolutionary changes that could or have occurred. With this, we could also gather a greater understanding of function based off of structural differences or similarities between unrelated proteins. This technology could also be used as a screening tool for pre-drug trials that could help create new treatments that would previously be unknown without knowing the structures or interactions that allow them to function. Even with the major advance that is AlphaFold, there are some drawbacks. For example, this method, while highly accurate, is only considered predictive and would still require validation using traditional methods. Another issue with the software is the fact that, while highly accurate when dealing with static structure, it is not as accurate when a protein has both an active and inactive structure. Conformational changes in folding are difficult for the software currently due to the dynamic nature of proteins and the variety of factors that go into folding beyond simple amino acid sequence. Similarly, folding shapes do not necessarily determine substrate binding, meaning that any possible drug predictions from the software still also require traditional methods to determine possible functionality. Thanks for listening and sticking around this long. I hope you've enjoyed learning about AlphaFold and the wonderful world of cutting-edge machine learning that comes along with it. If you're interested in reading more on it, here are a few different places I went to learn about AlphaFold. Have a great rest of your day.